Welcome. It may have been lengthy, but it was also generous. And I want to congratulate the Board of Trustees and especially Dr. Rahman for the creation of this remarkable institution and your dedication to high scholastic values and to the liberal arts as well as to the sciences. You've heard a lot about me. I'm not going to live up to my reputation, but I'm sorry your expectations are now unnaturally high. But what I will try to do is to tell you a little bit about my scientific career, but mainly to talk about cancer and to tell you how the kinds of discoveries that have been made by me and my colleagues that were already described to you by Rita Joseph have been influencing our attempts to control cancer more effectively. And I will end with some comments about trying to deal with cancer in the developing world. And well, I'm a scholar in residence. I'm in residence for a very short period of time because my flight departs at 9 o'clock. So, you've heard a fair amount about me, and I'm going to go over this fairly quickly. I come from immigrants. My grandparents came from Europe in the early days of the 20th century, and though they were relatively poor, they were able to send their kids on to college, and my parents were able to become professionals, a physician and a social worker, and allowed me to explore many aspects of human culture, and allowed me to become a scientist at a fairly late age. And I think while it's important for people who are committed to science to seize that excitement at an early age, for others of us, it's more appropriate to spend a lot of time thinking about what you might want to be and to have what I like to call a prolonged adolescence. Indeed, for a variety of reasons I don't have time to summarize, I didn't actually do any serious laboratory work until I was 28 and worked with the guy. Can we turn the lights down in the front? I think it's going to be hard to see the slides otherwise. But the guy on the left in the lower picture was my mentor at the NIH. Is there a storm? And as you've already heard, I did some work on the lactoferrin and bacteria, and I'm not going to go into this, you've heard a little bit about it, but the main thing was learning at the time before the recombinant DNA technologies became so commonly used. They weren't actually invented at that point, that it was possible to use some new tools to understand not just that we have genes, and the genes are encoded in DNA, but that genes are regulated, and that the regulation can be influenced by outside activities, and that the reason that one cell is different from another and why one cell is different in different places or at different times is because cells do respond to external influences by turning genes on and off, and that turns out to be an important aspect of the development of cancer as well. Now, history shapes our lives, and in a new country like Bangladesh, that's an obvious thing because you've gone through dramatic changes in your society over the last 60 or so years. The US is an older country, but nevertheless, in my own life, there were important challenges that were created by political and indeed technological developments. In my own life, when I was a college student, as some of you are here, the Soviet Union sent a satellite into the air to circulate the Earth for the first time, and the appearance of that first satellite, called Sputnik, changed the way in which the US viewed the development of science and technology. Science and technology were not just tools for trying to understand nature, there were also political tools that influenced a nation's reputation. And one of the ways to meet that challenge was to invest more into science education, into the support of science. Indeed, just a few years earlier, President Roosevelt had determined that because science had been so influential in the conduct of World War II, with the mass production of penicillin to treat wounds, the development of radar, the development of better anti-malarial drugs, the development of the atomic bomb, that it was important that the government spend money to encourage scientists to do work at the institutions around the United States, creating a system of government-supported science that allowed the country to do basic studies that then were converted into practical applications by industry and by contractors of various kinds. The second issue that influenced me quite a lot was the war in Vietnam, a war that I personally opposed, as did many of my colleagues, but that created a draft, and in other words, all physicians were required to serve in some aspect of government life, and I was, because of that edict, was able to serve my country by doing research, learning to do research at the National Institutes of Health in the last couple of years of the 1960s, providing an exposure to a way of life that has been my major occupation in the subsequent 45 years. Another important event, pictured at the bottom, is the signing of the National Cancer Act by President Richard Nixon. At that time, Nixon and most people in the country thought of cancer as being a fairly homogeneous set of diseases that affected many different organs. And the goal under the National Cancer Act was to expand funding available for cancer research and to try to control cancer within the next 25 years. Now, we haven't succeeded in doing that, but the stimulation of research by the passage of this bill did have a major effect on the way in which the cancer research was conducted in the US, expanding our resources greatly and allowing rapid progress of the kind that I'll briefly describe for you. The importance of having political leadership to support scientific research is critical in every country that I've visited to the development of strong scientific communities. And getting inspiring and meaningful public statements from our political leaders, especially in countries where science is supported to a very significant extent by the government, is critical. And indeed, near the end of this talk, I will show you our current president making an announcement about a new way to think about medicine, including the practice of cancer care, oncology, when he announced his precision medicine initiative, an effort to recognize the scientific changes that have occurred since President Nixon announced the National Cancer Act, a change in perspective that reflects the much deeper way in which we've come to understand the origins of the disease, especially cancers, by understanding the genes that cause cancers, the kinds of mutations that affect those genes and make them cancer-causing, and in a way that changes our perspective entirely on how we prevent and treat diseases like the various cancers, which we now know to be very different in all of their manifestations. Now, back in the early days, we knew relatively little about how cancer arises, but we had some simple concepts. We knew that normal cells can undergo growth and cell division. They can undergo a process of development that we often call differentiation. We learned a few years later that cells also have a means to kill themselves and be removed from the body. Aberrations, alterations in any of these processes can lead to excessive numbers of cells, a tumor, a growth of a tumor, and spread of tumors to other sites. And that may result from excessive growth, a failure to differentiate, a failure to undergo cell death. But we had very little idea beyond this general concept of how these changes might occur. The best route to trying to figure out how these changes might have occurred in the absence of the kinds of tools that are available today for studying cells and studying their genes was to make use of viruses that were known to cause cancers in animals. The virus that I and my colleagues chose to work on is a virus that was discovered in chickens over 100 years ago by a man named Peyton Rouse working at the Rockefeller Institute in 1910. And the important thing about this virus is that it had very few genes, and it was clear that at least one of those genes might be important in causing the cancer that resulted when that virus infected an animal. To make the study of that virus simple and amenable to biochemical and molecular tests, it was important to have a way to study the virus outside of the animal, especially in cells that could be grown in a petri dish. And about 47 years after Peyton Rouse's discovery, Howard Kemen developed a way to look at the process of conversion of a normal cell to a cancer cell, and that's shown in the lower right, where you see a pile of glittering cells that result from one infection of one cell by one virus particle, changing the behavior and the appearance of the infected cell. This is cancer in a test tube, basically, and it allows scientists who are quantitative in their approach to try to understand how the virus causes cancer. Now, a great deal of work at that point went into attempts to understand the small numbers of genes that are carried by the Rous sarcoma virus in its we call its genome, its full collection of genes, its chromosome. And this slide summarizes a great deal of information that I don't have time to explain how we came to know, but what it shows is that the Rous sarcoma virus chromosome had at least 3 genes shown on the left-hand side of the genome that are required for making new virus particles. But it also contained a gene shown in red, the viral Src gene, that is required for both changing the behavior of a cancer cell, of a normal cell, and turning it into a cancer cell. And you have to maintain that gene in the cell as an expressed gene, making RNA and protein, in order to maintain the cancer-like properties of the cell. Now, there were mutants of the Rous sarcoma virus that lacked the transforming gene, could still grow, but didn't change the behavior of cells after infection. And it was possible to use the information that was deleted in the formation of that mutant to do studies that revealed something very surprising, that the gene carried by Rous sarcoma virus to cause cancer in a chicken cell was a gene that had originally been found, a gene that was found in a normal cell from a normal chicken, and indeed a gene that was conserved throughout evolution of multicellular organisms. That gene was called the cellular Src gene. It turns out to differ in some very subtle ways from the viral gene. So the viral gene is a mutant form of the cellular gene. The cellular gene has an important function for normal cells, but it can be converted by mutation into a gene which has inappropriate biochemical properties that drive the cancerous change that we find in a cell infected by Rous sarcoma virus. Now, that provided a new way of thinking about cancer. One could imagine that while the tumor virus, Rous sarcoma virus, is a useful experimental tool, it also carries a gene that introduces us to a normal cellular gene which has the potential to become cancerous in the normal cell by undergoing mutation. And that general idea that there are genes, we call them proto-oncogenes, precursors to viral oncogenes, that might be the missing link in trying to understand how factors that place us at risk of cancer, like the chemicals present in cigarette smoke, or radiation, or other chemicals in the environment, might be causing mutations in genes like the cellular Src gene, causing cancers in animals and, importantly, in human beings. Now, this story became somewhat more complicated as other viruses related to Ralph sarcoma virus, other so-called retroviruses found in not just chickens, but also mice and rats and even primates, subhuman primates, were studied. And a large number of colleagues in this field of cancer research discovered such genes as viral oncogenes and traced their origins to cellular genes, which we call cellular proto-oncogenes. And it became apparent with time that many of these genes were actually mutated in human cancer. So the general idea that we could find the genes that play a role in the causation of human cancer by using retroviruses as experimental tools turned out to be correct. Now, there were other ways to try to validate this idea, and one way, espoused in part by my colleague Robert Weinberg at MIT, was to try to identify the mutant genes in a human tumor by using the DNA from a human tumor to try to cause cancerous changes in normal cells in culture, just by taking DNA from a cancer cell. And when he did that, he was able to identify cells that had been changed in their behavior to a cancer-like state, and then by doing a series of molecular procedures, he was able to show that the genes in the human tumor that were responsible for the change in cell behavior were indeed very commonly the genes that we already knew about from the study of retroviral genes. And one particular class of such genes that I'll return to in a moment are the RAS genes. Now, I want to emphasize one thing in all this science. I know some of you are not absorbing all the scientific details here, but I want to emphasize that the work that gets done in laboratories like our own is a product of teamwork. I have an unusual relationship with my friend Michael Bishop, with whom I worked for about 20 years. We're shown here in various places as working scientists at a meeting at Cold Spring Harbor Laboratory in the 70s, as recently announced winners of the Nobel Prize in the late 80s. Our larger team is shown in the dinner party down in the lower left, including my lovely wife on the right. She was part of the team too, in a different way. And then the congenial and the sports-oriented aspect of our lives is shown down the lower right, where we frequently got our laughs together to play softball. Now, I'm going to shift to a slightly faster pace chronologically by telling you that while over 20 years we were able to develop a notion of how cancer might arise by mutations affecting a class of normal cellular genes known as proto-oncogenes, might lead to the development of cancer. But at the same time, as we were doing this work, people were trying to develop new methods for looking at human DNA much more directly by simply chopping it up into pieces, isolating each piece, and determining the nucleotide sequence of pieces of the human genome and putting that all together. And I'm sure everybody in this room has heard about the Human Genome Project, which by 2003 allowed us to see the complete expanse of the roughly 3 billion units of human DNA spread out over 23 pairs of chromosomes. The techniques that were required to do this monumental task in the history of human knowledge can be applied quite readily to the analysis of the complete set of genes in a human cancer. And in the course of the 2000s, and starting about 2005, a couple of the institutes at the NIH put together a project called the Cancer Genome Atlas, TCGA, standing for the 4 letters of DNA, to look at a large number of types of tumors, many different examples of each one, using a large number of characteristics, some clinical, some molecular, and to try to understand in much greater detail the nature of individual cancers. That's required a team effort on an unprecedented scale with the development of teams to characterize the tumors that were being studied, to determine the complete nucleotide sequence of many parts of these cancer genomes, and then to analyze those genomes, looking for patterns that would help us in the future diagnosis and treatment of cancer. The result of that extensive study, which I don't have time to describe in detail, was to produce a picture of cancer that's much different from what we had at that point. It became clear that cancer genomes were very complicated, that there were mutations we could recognize as being important in the development of a cancer, but many additional mutations that probably played either minor roles or were simply passengers along for the ride. And that made the analysis more difficult. We also recognized that some cancers had very similar mutations in every example of the cancer, but other cancers were very different from one individual to another. We also recognized that many of the mutations that were important in human cancer affected enzymes against which it was possible to develop drugs that had the prospect of being very beneficial in the treatment of cancer. And this notion of precisely identifying the mutations responsible for each cancer and then finding drugs that could be used to treat those cancers gave rise to a concept of targeted therapy that is obviously much more rational than conventional therapy with drugs that were often referred to as chemotherapy that simply kill cells that are dividing excessively. There were reasons to think that targeted drugs might be effective. We knew from work with animal viruses that cancer genes were required not just to initiate a cancer, but to maintain it. And we knew this was true in both cultured cells and mouse models. And we learned quite quickly that cancer genes were required for human cancer cells to survive as well. So let me show you the classical example of how that works. A disease called chronic myeloid leukemia affects about 6,000 patients in the US every year, roughly 20 times that many people worldwide. Virtually every patient with the disease has an abnormal chromosome that's made by recombining a part of chromosome 22 with chromosome 9, creating a hybrid or fusion gene in which one of the oncogenes discovered by studying retroviruses, the so-called ABL gene, is fused with something else. And the result is an abnormal protein that has an uncontrolled enzymatic activity. A drug called imatinib, or Gleevec, its commercial name, was developed and shown to inhibit the enzyme that's present in the abnormal fusion protein that's present in all patients with chronic myeloid leukemia. When that drug was used to treat patients with chronic myeloid leukemia, virtually every patient showed a rapid regression, that is, a rapid disappearance of virtually all aspects of the disease. A few abnormal cells remained behind, but these patients were basically healthy once they started on the drug. Sometimes they developed resistance to the drug. That's another problem for another day. But additional drugs were developed that overcome the resistance. Moreover, the drug could be taken orally for very long periods of time, and patients who had this once lethal disease now have the prospect of a normal lifespan. In addition, it turns out that a few other mutant enzymes respond to this drug, and it means that some other generally fairly uncommon cancers can be treated with this drug as well. So this was a major development, and it gave proof of the validity of the basic idea that targeted therapies might work. But we shouldn't be misled by this example, because while chronic myeloid leukemia is a reasonably common cancer, it's 6,000 cases per year in the US, it's very far from being the predominant form of cancer. The US has well over 1 million cases of cancer a year, and for most of those cancers, we don't have a therapy that is quite as effective as this. And there are several reasons why this is true. there are many mutations, not just the one I've described for myeloid leukemia, present in most cancer cells. There are many different kinds of genes that are mutated. Not all of them are as easily inhibited as the abnormal ABL protein. There is an ongoing mutational process in cancers. Cancers continue to evolve genetically, creating ways to develop resistance against targeted drugs. And all those things have made the advent of targeted therapy an encouraging development, but very far from being the final solution to the problem of human cancer. So there are lots of reasons why the cancer's complexities have interfered with the success of targeted therapies. I'm sorry, this is a little screwed up because I had to move this onto a new computer, but bear with me. An important feature here is that some of the targets that we would like to use as targets for therapy are very difficult to interfere with. And a common one is one that I mentioned earlier, the RAS gene mutations. I've mentioned those in the context of a couple of previous experiments, but they're especially important because they're extremely common in human cancers. Virtually all pancreatic cancer, which kills 40,000 individuals in the US every year, have mutations in one of the RAS genes. About half of colorectal cancers, about a third of one kind of lung cancer, and other cancers as well. And therefore, targeted therapies against these mutations would be highly beneficial. But it's hard. There is a biochemical reason I'm not going to have time to explain. RAS proteins, in a three-dimensional sense, are very difficult to treat with drugs because they have a very smooth surface without niches and crannies where drugs can reside. And the way in which RAS works in the cell is very complicated and hard to interfere with. So it's been difficult to generate drugs that are targeted against these very common mutations. For that reason, over the last few years, when I was at the National Cancer Institute, we developed a new RAS initiative that's brought together hundreds of people from around the country to try to find new ways to treat cancers that have mutations in this class of genes. It's also inspired us to think about other kinds of solutions to the problem of the inadequacies of targeted therapy. I'd like to conclude with just a few of those. First, we're all focusing now not just on the individual mutations that I've described in a little bit of detail, but also on the underlying patterns of mutation, the pathways that are affected by cancer mutations, and changes such as changes in cell metabolism and other aspects of cell growth, your ability to invade and to spread to distant sites. It might be better targets for new kinds of therapies, not just the proteins made by individual mutated genes. Second, we need to think more about the responses of the host that are common to many cancers, and not just focused on the genetically damaged cell, which is variable from one patient to another. And I'll come back in a moment to the question of emphasizing public health measures. So let's just say a couple of things about these host responses. We now know and recognize more and more every day that, well, the cancer cells themselves are obviously critical to the pathological process of cancer, but these cells do not exist on their own. They're surrounded by blood vessels that are created with normal blood vessel cells. They're surrounded by other kinds of normal cells that are sending signals back and forth to the cancer cells, and they're frequently invaded by immune cells that represent a host response to the presence of cancers. Cancers have mutant proteins. They are recognized as being at least partially foreign, and the immune system is therefore able to set up a response to those cells, which in the past has never been adequate, as far as we can tell, for elimination of the cancer. Over the last several years, it's been possible to take advantage of our increasing knowledge of the immune system to try to treat cancer more effectively. Some of this is as simple as making so-called monoclonal antibodies against proteins, even normal proteins that we know to exist on the surface of cancer cells. It's been possible to design some abnormal proteins that contain toxic elements that kill cancer cells. It's been possible to do genetic engineering on one component of the immune system, the so-called T cells, to force those cells to attack cancer cells. And most importantly, in the last few years, it's been possible to make antibodies that inhibit an aspect of the immune system that was only recognized recently, namely the ability of the immune system to inhibit its own reactivity. By using antibodies that block the components that mediate inhibition of the immune system, it's been possible to activate the system more rigorously so that it now attacks cancers. And indeed, in a small number, but a significant number of patients with certain kinds of cancer, melanomas and lung cancers, a few patients with colorectal cancers, these new inhibitors of the immune checkpoints have been able to place patients into a sustained remission that's lasted for several years and may be permanent. Now, this should not be happening. Okay, so all of these things that I've been describing, targeted therapies, a precise definition of the genetic changes in cancer cells, the use of new tools like the tools of the immune system, have been inspirational to our country's leader. And a couple of months ago, when I was still at the National Cancer Institute, he brought a number of us into the East Room of the White House and made an announcement about what we call precision medicine.
I sent this to Congress on Monday will include a new precision medicine, and all of us access potentially to the personalized information that we need to keep ourselves and our families healthy. You can match a blood transfusion to a blood type. That was an important discovery.
President Nixon's declaration of a battle against cancer as one disease, and contrast that with what President Obama was telling you, which is that it's important to tailor our therapies to the differences between cancers arising in different organs, cancers arising in the same organ in different individuals, recognizing that genetics has taught us how complex cancer is and the need to be much more precise in the way we diagnose and treat this disease. Now, this is not an entirely new idea, and indeed the Institute of Medicine in the US had envisioned the development of this kind of change in the way we think about disease by imagining something that is central to this whole process, namely building large collections of data that allow us to extract information that's informative about diagnostic categories and new ways to prevent and treat disease. And in the center of this diagram is that computational network based on modern computer science, illustrating the critical nature of multidisciplinary work in the sciences these days. We cannot function as medical scientists without the chemistry that produces fast DNA sequencing, without the computer science that organizes that information in databases that we can manipulate, and requires the use of clinical tools to make precise observations and other kinds of biomedical research, all of which feed back into this information commons or knowledge network. And this is a challenge that engages not just physicians and molecular biologists, but also computational scientists as well. I want to say finally a couple of words about public health, because we focus largely on improving the diagnosis of cancer by understanding its genetic basis and changing the way we do cancer therapy through targeted therapies or immune therapies, to emphasize another part of the cancer dilemma, which is how to prevent it more effectively and how to bring the changes in our approach to cancer to parts of the world that have fewer resources, both personnel and financial resources, to combat a disease as complex as cancer. And indeed, traditionally, cancer has often not been thought of as part of the global health mission, in contrast to infectious diseases, because it's often thought to be too difficult to prevent or treat cancers in the developing world. And my colleagues and I at the National Cancer Institute disagree with that. We think there are many things that can be done, and there is both a growing need to confront cancer in the developing world and a number of fairly inexpensive, effective approaches that are feasible to introduce even in the poorest countries a great deal poorer than Bangladesh. The need here is dramatic because we know that with an increase in life expectancy and a decline in other causes of illness and death, particularly infectious disease, that overall cancer deaths per year are going to rise, and that increase is going to occur in the lower and middle income countries. And that pattern is already fairly well documented in countries that have appropriate cancer registries and are documenting the increase in frequencies of cancers. So we developed at the National Cancer Institute just a few years ago a Center for Global Health that is operating in a number of countries, not at this moment in Bangladesh, but in India, China, Mexico, Turkey, Indonesia, and other places, bringing a variety of measures to the efforts to control cancer more effectively in those countries. That includes finding out what kinds of cancer are actually present in those countries through the development of accurate registries and the development of national cancer plans to focus on the infections that are associated with cancers, especially the use of vaccines against the human hepatitis B and the human papillomavirus, both of which are causes of major kinds of cancer and for which vaccines exist. We try to work with developing nations that are attempting to make improvements in cancer control by developing improved screening, access to treatment, access to palliative care, and improved surgery. We try to emphasize the importance of using tools to reduce tobacco use, to minimize obesity, to reduce the use of alcohol. All these factors being important not just in the incidence and mortality due to cancer, but also for the control of cardiovascular and lung diseases, and in some cases metabolic diseases like diabetes. And finally, we've been interested in trying to build capacity in developing countries by taking advantage of the enthusiasm for improving global health that's present on our campuses and in our cancer centers in the US, and to find willing partners in developing countries to try to pursue some of these goals. So the battle against cancer is a big one. I've tried to summarize how work on the basic genetic underpinnings of cancer have led to changes in our concepts of how to treat cancers with drugs. Our understanding of the immune system is bringing new kinds of tools into cancer therapy. How we are trying to think about ways to overcome the complexity of cancer by focusing on commonalities of cancer and by making better use of public health tools to try to reduce the growing tide of cancers that are going to be affecting public health even in the poorest parts of the world. Again, I want to thank you for your attention here today, and